Hello, thank you for this opportunity to give this seminar for Indiana University. My name is Raina Plowright. I'm an associate professor of epidemiology and I'm at Montana State University. I'll tell you a little bit about myself as I get started here. These bats you can see hanging in the trees are Pteropus medius. They're the major reservoir for Nipah virus for Bangladesh and for India. And they're one of the species that we actually study in the field in our studies of emerging infections. So I'm going to be talking about a few topics today, but mostly focused on pandemics, how they start, and how we can prevent them from happening. I lead a large group of collaborators, which is a transdisciplinary research group working on pandemics, trying to establish the scientific basis for pandemic prevention. We have about 70 people. We're dispersed across 5 continents. We have field projects in Bangladesh, in Ghana, Madagascar, and in Australia. And we're especially focused on the pathogens from bats. This is a Pteropus electo. It's the major reservoir of Hendra virus in Australia. Hendra virus is another Nipah virus like Nipah virus, and it's having a drink of mango juice here after we've just given him an anesthetic and taken a blood sample and a urine sample. These are amazing creatures, bats. There are over 1,400 species of bats, and they're now being associated with many different emerging infections. A reason we think they may be good reservoirs of emerging infections is because they are the only mammals that fly. And that gives them special adaptations, which we think are important in allowing them to suppress their viral load, or so to cope with these infections without actually getting sick themselves. They're also very unusual creatures. For example, this is a Brand's bat. It is around 40, this bat has been recorded at 41 years of age in the wild. It weighs around 5 grams, so really the weight of a nickel. And that ability to have a long life without going into senescence, despite having extremely high metabolic rates required for flight, is also one of the unusual features of bats, which we think is tied up with their ability to host emerging infections. So flight is really critical. We're still trying to understand this. We have many theories and very few of them are actually proven, but certainly something about bats is really critical. And I want to say, bats are not the bad guys here. Bats offer these extremely important ecological services: pollination, seed dispersal, insect consumption. These are ecological services that no other species can provide. I think bats are more the canary, the sentinel. They're an indicator of our changing relationship with the environment, as opposed to this villain that is a reservoir of emerging infections for humans. So let's start on how pandemics start. To have a pandemic, these 4 processes have to occur. We have to have an infection in a reservoir host, infected reservoir hosts. And the kinds of factors that determine which reservoir host is a species that which populations maintain an infectious disease. To be able to maintain an infectious disease and then to be able to shed that pathogen into the environment or into a situation where transmission can occur requires complex dynamics of those infections. So if you think about COVID within human populations, and you have some populations with soaring infection rates and some with a trough in infection rates, those same dynamics are going on in wildlife populations in space and time. And we study the infections in wildlife populations in the same ways that we study human populations. So for a pandemic, we have to have the infected reservoir host. That reservoir host has to release the pathogen from the host. In this figure here, it's through shedding, so excretion of the pathogen. Here is a bat excreting a virus in its feces or its urine. We know the Nipah viruses are excreted in urine, the coronaviruses in feces. We then have to have a spillover event. And spillover is the passage of a pathogen from an animal to a human. It's actually incredibly complicated. It's not just a quick event. There's a lot of things have to align for that to happen. And then for a pandemic, there needs to be spread then within the human population. I'm going to introduce this term land use induced spillover. This is where we understand most of these spillover events from wild animals to humans. They happen in the context of landscapes. And land use change is often the trigger of this cascade of processes that leads to a pandemic, this infect, shed, spill, spread cascade. And then another term I'll introduce is landscape immunity. This is the ecological conditions that support the health of species, these wildlife reservoir hosts in situ, that dampen prevalence and all of the situations that would lead to high rates of disease and proximity of animals to humans, allowing spillover. And we have a preprint in review here that you can look at to see a broader definition. So infect, shed, spill, spread. As I said, it has to happen for every pandemic. How about for SARS-CoV-2? How are these processes involved here? Well, we had to have an infected bat. We know that it's the Chinese horseshoe bats are the most likely reservoir hosts. SARS-CoV-2 hasn't actually been identified in its reservoir hosts, but similar viruses, so recent ancestors to SARS-CoV-2, have been found in horseshoe bats in this region here, Yunnan province, northern Vietnam, Laos, this northeastern part of Myanmar. So at some point, an infected bat in this area here was either transported to Wuhan or infected either a human or another species in this region, and that human or other species was transported to Wuhan. But infect, shed, spill, and spread all occurred, and it occurred in an area where there is high rates of land use change, high rates of forest fragmentation and deforestation, high human population density, and high density of livestock species. So all of the conditions that we think promote this process. How do we predict pandemics? Well, if we look at coronaviruses, we've had 3 coronaviruses from bats spill over into human populations in the past 20 years. The first 2, SARS in 2003, and then Middle East Respiratory Syndrome around 2012, have come through an intermediate host, civets for SARS, camels for MERS. And in camels, actually, the spillover to camels happened probably decades ago. We don't know for SARS-2. But what we do understand is that all of these 3 viruses probably originated from a single spillover event from a bat into whichever was the next species in that chain. So we essentially, to understand spillover of coronaviruses, we have n equals 3. And that's not possible then to try to build the patterns and understanding of how spillover happens so that we can then start to work on how to prevent it. And we have many, many more coronaviruses out here. Here is a map produced by some members of my lab who have looked at the different subgenera of coronaviruses, and you can see the colors across the map. Coronaviruses are widely distributed. There are more than 3,000 variants in bats. More than 200 of them are SARS-like, and so potentially zoonotic. So trying to prevent the next coronavirus pandemic is going to be very difficult given the limited information that we have. So I suggest then, why don't we look at systems where we do see spillover, regular spillover, a lot of spillover, so we can start to try to understand the patterns and processes that lead for those events that spill over and spread within humans. So I'm going to look now to Nipah virus in Bangladesh, which is Nipah virus, this is the genus Henipavirus, family Paramyxoviridae, and Hendra virus, also in the genus Henipavirus, family Paramyxoviridae. This is Bangladesh. You can see here we've got latitude and year. Every dot there is a spillover event from a bat to human that caused either one or more infections in humans. In Australia, it's the east coast of Australia. So we have space, we have time, and every dot here is a spillover event from bats into horses in Australia. Some of these events involve multiple horses as well. So we have now two systems where we frequently see spillover. We see spillover every year, and that can allow us then to try to understand this process. Again, this is Pteropus medius. This really beautiful large fruit bat, or we call them flying foxes as well, in Bangladesh. And this is the type of situation which we are really concerned about these viruses spilling over into. We see spillover every year in Bangladesh. It is the most densely populated nation that isn't a city island. And Nipah virus is a WHO top 10 priority pathogen, meaning that it's one of the ones that we're worried about generating a pandemic and for which there are not any countermeasures in place right now, no vaccines or therapeutics. Every time Nipah spills over in Bangladesh, it's a roll of the dice as to, do we get the right genotype that can start to spread within humans? So far, all of the transmission within Bangladesh has been short chains of transmission. So the reproductive number of this pathogen in humans is less than 1. But we know that there are numerous varieties of hennipaviruses from sub-Saharan Africa across Asia, Oceania, and Australia. Almost all of them have not been characterized. So we don't know whether there are hennipaviruses that can actually spread. So let's look at how spillover actually occurs with these viruses. In Bangladesh, it's through date palm sap collection. So guys will climb trees, make a gash in the tree and put a little straw in that drips the date palm sap into the pot. And then it's sold on the street, usually after collection overnight. And during the evening, animals like, this is Pteropus medius here, our fruit bat, come and drink the sap as it's coming into the pot. And here you see, you just saw it turned upside down. Here, I think I'll just show it again, turned upside down and peed right in that pot. Then that date palm sap is sold on the street the next day. And you can track the outbreaks of Nipah virus according to the roots of the date palm sap sellers. So here you see a red line. This is the root of one date palm sap seller, and the dots on either side are cases of Nipah virus that we believe were caused by this date palm sap collector. And Nipah is a highly fatal disease. Each outbreak has a fatality rate between 50 and 100%. So it's a devastating pathogen. Now let's jump to Australia, where the cousin of Nipah virus, Hendra virus, is, and Pteropus alecto being the major species that is a reservoir host. It's a flying fox or fruit bat. We see spillover of Hendra virus when these bats come into trees in feeding paddocks where horses are grazing, and they excrete virus on the ground, and horses then consume that contaminated grass. Its fatality rate in horses is around 80%. They get very sick, and then people only get infected through horses. And usually it's veterinarians, it's caretakers, people who are looking after sick horses who are infected. Hendra virus has changed the veterinary industry on the east coast of Australia. We now understand that any horse that has a fever, neurological disease, respiratory disease, actually even a colic, tummy ache, could have Hendra virus. And it has a 60% fatality rate in humans. There have only been 7 cases in humans, and each one of those has been devastating. And even though we don't see the large numbers that we've seen cases for Nipah, Ebola, and coronaviruses, this disease has had a terrible impact on the equine and veterinary industry in Australia. Another thing about Hendra is it's occurring in a country where we have a very good science infrastructure and we have long-term data sets. So it's becoming the model system for understanding bat virus dynamics. And now we're using our insights on Hendra to transport them to what we understand about coronaviruses and Nipah viruses and hennipaviruses in other parts of the world. So what we understand about Hendra is that we see spillover events nearly every year, but we see clusters of spillover with interannual variability. So we had a large cluster of spillover on the east coast of Australia in 2011, then another one in 2013, 14, and in 2017. I'm going to show you something from this 2017 cluster. Also, just to note, this is Brisbane here. So we're at the top part of New South Wales and the bottom part of Queensland in Australia here. After that outbreak in 2017, we went to visit one of the properties where a horse had died. My group had just been awarded a National Science Foundation Coupled Natural Human Systems grant, and we were studying the series of events that lead to spillover events. And we're talking to the horse owner. And she said it was raining when her horse was killed by Hendra virus. It was raining, and because it was raining, her horse's paddock had turned to mud. So she moved her horse into the orchard. She also said that a new population of bats had just formed in her neighborhood, about 1 kilometer away. And they'd not seen bats in the area before, but now there was this new population. And she said that the bats were coming in to feed on the lemons in her orchard, the same orchard she'd put her horse. Now, we understand from our data and monitoring at that point, the bats were experiencing a severe acute food shortage. Lemons are a food of last resort. They're a starvation avoidance food. So if the bats had not been suffering a food shortage, they wouldn't have come into her paddock to feed on this resource of last resort. And if the bats hadn't been going through this food shortage starvation event, they probably would not have been shedding Hendra virus. We understand there is a link between nutritional stress and Hendra virus infection in bats. And if it hadn't been raining and cold at that point in winter, when bats excreted the virus on the ground, it probably wouldn't have survived for very long. But cold, moist, wet conditions are actually really ideal for viral survival. And if all of those events had not lined up, then we wouldn't have seen her horse be killed by this terrible virus. And then she wouldn't have gone through the most stressful time in her life, where her family was potentially exposed, but thankfully we're okay. And her pets were quarantined inside for weeks. Her whole property was quarantined. But while we stood there in the paddock, we're actually standing in this lemon paddock as she explained this story. It was an amazing experience as a scientist, because we'd just published this conceptual overview of spillover, of all of the things that have to come together for a pathogen to go from an animal to a human or another species. In this case, it's a horse. And it just was like everything sort of came together into place here, that we had to have everything has to line up in the right place. Every barrier has to be overcome at the same point in space and time. So we had the bats change their distribution to come near into this agricultural area where there are horses. We knew the bats had a high prevalence of infection. We actually were sampling the bats at the time. The bats were shedding the pathogen. We think these bats are maybe persistently infected, but only excrete the pathogen when they're stressed. So they're shedding the pathogen. The survival of the pathogen was ideal. And then there's a whole bunch of within host barriers within here, horses, that also had to come together. So this whole cascade of infect, shed, spill, spread had really come together at this one paddock in Eastern Australia in 2017. So we actually predicted that cluster of spillover events in 2017. We'd put out letters to the Australian Veterinary Journal. We had warnings from this chief veterinary officers that there was going to be spillover. And we did that because we had these indicators now from long-term data of bats on the east coast of Australia that there was going to be a food shortage then, and that would lead to clusters of Hendra virus. And we're able to do this with actually machine learning algorithms. We were able to show that if we have any of these indicators of a food shortage, that we're likely to get a cluster of spillover. So bats coming into rehabilitation centers, bats with poor reproductive success at the end of the previous year, and a high oceanic Niño index, which is an indicator of a high temperature of the Pacific Ocean. We actually saw these same conditions coming into 2020, and we put out the same warnings. We had warnings for veterinarians and horse owners across the east coast of Australia. And this time in 2020, in 2017, we were right. Actually, the spillovers started happening the day our letter was published in the Australian Veterinary Journal. In 2020, we were wrong. And we now understand why we were wrong. And it's one of these times of serendipity in science. The reason we're wrong actually offers us a great amount of hope in actually being able to stop this system from having spillovers. And I'll get to that right at the end of my talk. First, I just want to talk a little bit about the ecology to understand why we think these spillover events are happening in Australia. These bats, flying foxes or fruit bats, they're nomadic. They're nomadic nectivores, and they aggregate in large populations, and they feed in forests. And forests have these ephemeral, patchy flowering events where a whole forest might flower, that might last a month or 2 months, and then the flowers are gone and the nectar is gone. And so to be able to get a constant supply of food, the bats constantly move across the landscape in these huge aggregations of, it can be tens to hundreds of thousands of individuals. Certainly historically, it was hundreds of thousands of individuals. So now I'm going to show you the food supply. So the forests that actually produce those flowers and the nectar for these bats. In summer, most of this forest is in montane areas, which are very well protected. So mountainous areas of Australia that are very well protected. This is work by Peggy Eby, Dr. Peggy Eby, who's a bat ecologist in Australia, who's amassed these incredible data sets to put this story together. So this here, you can see there's much flowering habitat available in summer. Then if you look to winter, there's very, very little habitat. And most of it is in lowland, flat coastal areas that have almost been entirely cleared for agriculture. And there's only a handful of species of trees that even provide nectar in winter. And in many of the places where we work, it's almost entirely gone. So 96% loss of winter habitat in some of the areas where we're working. What we think has happened now is that we've had a combination of loss of winter habitat and these natural cycles of El Nino integrate or interact to cause these food shortages. And historically, these food shortages always happen. We've always had climatic cycles. The bats always go through periods of time where there may be a brief nectar shortage. Usually it used to be just a few weeks, so not even captured really in the data we have. So these nomadic populations during a food shortage, they would split up into small populations, and they would go to find reliable food, usually fruit. Historically, it was probably rainforest fruit, but now it will be urban fruit in urban or agricultural areas, like fig trees planted for shade in horse paddocks. But as soon as that food shortage abated, the bats would then re-aggregate into their big nomadic populations and start feeding on nectar again. But what we've been seeing, particularly over the last 15 years, is that this fissioning process is happening, but we're not seeing this return to nomadism. And we're seeing these small populations of bats now sticking in agricultural and urban areas. And it's these populations that are responsible for spillover to horses, just like that new population that had moved into the area near the paddock of the horse I described. What this looks like across the east coast of Australia, it looks like this proliferation of bat roosts. Every red dot here is an occupied roost. And you can see from 2002 to 2017, we've now got bat roosts in all sorts of places where they were not historically present. And if we look, this is the number of roosts over time. We see this proliferation of roosts is consistent across the east coast of Australia with these stepped increases that are associated with food shortages, severe food shortages. What that looks like here in our study area, which is this part of the subtropics of Australia, so southeast Queensland, northern New South Wales, is a tripling of the number of roosts. Flying fox roosts is the number of roosts since 1995, tripling of the number of roosts. And if we now look at hendrovirus spillover, the hendrovirus spillovers essentially mirror the pattern of roost fragmentation. So when we have a food shortage, we have a step increase in the number of hendrovirus spillover events. At the same time, we have a step increase in the number of roosts. A really big component of this, which I had just described, is the location, that these bats have changed their distribution from feeding in native forests historically to now they're feeding in places where horses are. And if we just look at the distance of bats to urban land use between 1998 and 2019, you can see by 2019, this distribution is really squashed here. So almost all of the bats are really close to some kind of urban land use. And what this looks like across the landscape here is each dot being a bat population, and the red is urban areas. We've gone from, and each of these is a feeding area of a population. You can see the number of small bat roosts here now throughout the urban areas in this part of Australia. So essentially we have bats now in contact with horses, allowing that infect, shed, spill process to occur more readily. And how do we know this? We know this by putting GPS collars. Here's a GPS tracking device glued to the back of a bat. And we look at where the bats feed. And we have this beautiful data from a PhD student in my lab, Maureen Kessler, showing that during food shortages, the bats are feeding in horse paddocks. And when there is nectar, abundant nectar available, they fly great distances to feed in native forests away from horses. But there's more to the story. As well as this contact process, we think that when the bats are going through these nutritional stress events, they're shedding more virus. And we think that there is a link between the immune status of the bats and their likelihood of shedding the virus. How do we know that? Because we work in the field. Here's my PhD student, Devin Jones, and catching a bat here in a net. So here it's very noisy. And I can tell you, it's very noisy and smelly. We catch the bats in nets as they're coming back from feeding. We untangle them. This is a crocus palm. It's an invasive species in Australia that the bats like to eat when there's not much native food available. He's not going to let go of that. Each bat has an anesthetic, and we take a blood sample from the bat. That blood goes into a bunch of studies on the immune system. We look at you can see an FDA card there for looking at the bacteria within red blood cells. We look at the energetic status of these bats, nutritional status. We take urine, and from the urine, that's where we find the virus, but we can also study the metabolism of bats from the urine. And then the best part is we get to release them back to the colony at the end of this procedures. And that's the Australian field crew there catching Tropus electo. And here you can see it's a public park. This is the kind of place where bats are roosting in these new urban areas, often parks, school grounds, creating a lot of conflict actually with the local communities. This is us working in the Northern Territory of Australia, where we weren't able to catch the bats in nets because there were too many crocodiles on the bank, and we were concerned the crocodiles learn your behavioral patterns. And so we had to keep moving our nets every day. And then we came up with this idea to catch from the boat, which was very, very successful. The bats would fly into the net, we'd be able to get the net down, and then we untangle. This is a little red flying fox, so it's a different species. It's a smaller flying fox. And we'll put the little bat into a pillowcase, and then it will go through the same procedures as those large Tropus electo, so anesthetic and blood and so on. But field work is a creative process, that's for sure. So what we do is we have 5 populations on the east coast of Australia, and we go back to them every month. Most of our work is done actually by looking for virus under the roosts in plastic sheets. We don't have to catch the bats for that. And then we also have 2 populations where we capture the bats consistently. And then we're also capturing bats in nomadic populations. So we can compare these new urban populations to the traditional nomadic populations. These are some of the results here. This is prevalence, the proportion of bats that have or excreting Hendra virus in their urine over time. And you can see that it's very variable over time. This is aggregated over our field sites. Here you can see it disaggregated here. But I just will show you this point here. This is the, that is the prevalence. Over 60% of urine pools were infected with Hendra virus at that population in 2017 near the horse that I had described. So we had a pulse of excretion at that point in time. We understand that when we see a pulse of excretion of Hendra virus, we're seeing many viruses excreted. This is work by Alison Peel we published last year, showing every color here is a different paramyxovirus. Here is Hendra, and here are all the other paramyxoviruses. This is during a pulse of excretion in 2011 after a severe food shortage for bats. I think if we start looking at other families like coronaviruses and filoviruses and other pathogens that could have zoonotic potential, we'll start to see that there is excretion of many viruses at once. And so far, our studies have only been limited to paramyxoviruses. So in summary, what we think is happening in this Hendra virus system is that we have an interaction between climate oscillation and loss of specifically winter habitat of the bats. This is leading to food shortages, which leads then to the fragmentation of small populations of bats that move close to reliable food sources. They're usually poor quality food sources, and hence it's an energetic decision the bats are making. If they're close to a poor food shortage and they don't have to fly very much, they can maintain their energetic status. Whereas with nectar, it requires a lot of travel, and they get so many calories from the nectar. Actually, there are stories of bats having to fly extra kilometers to try to burn off all of that sugar. But this population fragmentation into urban areas is an energetic issue. It's really these bats are in a low energetic state. When those bats in these urban agricultural areas then encounter a new food shortage, we see spillover. And we see that because they're feeding in horse paddocks close to horses, and we think they're shedding more virus at those points in time. How do we actually prevent pandemics then? Well, as I said, in 2020, we predicted a cluster of spillover events. We had a food shortage in bats, we had poor reproductive success, a big surge of bats coming into rehabilitation centers, and we had a high ONI, Oceanic Nino Index. And we put out a letter in the Australian Veterinary Journal warning people to vaccinate their horses, which we actually understand they probably didn't do. We had one Hendra virus event at the very early winter, and so we thought we were on track to have many spillover events because it was so early, but actually that did not happen, and I'll explain why. Firstly, I just want to say that there is vaccination for Hendra virus. It's effective. It's a good vaccination, and it's not being used. We didn't see in the data an uptick in vaccination rates after our warnings, but the vaccination rates are generally very low. It's for many reasons, partly because of anti-vaccination narratives about this, social media driven, very similar to the MMR vaccination. But if you think about vaccination and where on this hierarchy of pathogen spillover I showed earlier, where it's working, it's essentially reducing the susceptibility of that recipient host. I'm going to get back to that in a second. So why didn't we see spillover? Well, we had an unusual and unpredictable mass flowering event that happened in July, so in the middle of winter. So we had a June spillover event, and then in July we had a flowering event that attracted 200,000 bats to feed on blue gum in Queensland. This is the population before the flowering event, the extent of the bat population. This is the extent of the bat population after the flowering event. And what we think has happened is that when we do get these mass flowering events, they do stop pathogen spillover. And we look through the data historically. There's a huge stand of spotted gum that produces abundant nectar, but only every 4 to 7 years. And we've looked through the historical data, and every time there is a spotted gum flowering event, which is enormously productive and lasts really for most of the winter, there is no Hendra virus that winter. So what we saw this winter, it wasn't a spotted gum flowering event, but it was another large, unpredictable flowering event. And we've now gone back through our 16 data sets over 24 years, and Peggy Eby has been able to track the flowering events that have occurred over time. Using a Bayesian network model, now we've shown that food shortages lead to spillover, but winter flowering events stop spillover. So we now think we have a really sustainable, scalable solution to spillover. And that is to replant those winter flowering trees, replant the handful of tree species that can provide abundant nectar in winter. And essentially what this would do is would allow this system to reverse from this fission to nomadic. Just like when we saw this large winter flowering event in 2020, we saw bats empty out of those small populations, regain their nomadic lifestyle. We saw actually the majority of one species aggregated around that one flowering event. And so if we can do this, we can change this host distribution back to their native forest. If bats have this a good nutritional status, then perhaps we can reduce the prevalence of Hendra virus and shedding, and certainly the risk of exposure to horses. So in conclusion, we've been able to identify what we think is the root cause of zoonotic spillover in this system. And we've done it by amassing long-term studies. So we have 16 data sets over 24 years. Much of this is a result of one person, Dr. Peggy Eby, and her long-term monitoring of flying foxes on the east coast of Australia, and then this transdisciplinary team that we've been able to assemble to try to understand the virus, the bat ecology, the bat behavior, and the virus dynamics within the bat populations. And we think that by addressing the root cause and paying attention to this concept of landscape immunity, that perhaps pandemics could actually be preventable. And I'll leave you with the release of a little red flying fox. And thank you very much.